So we ended up enrolling 24 patients, 23 of which actually started therapy. And we just looked at the patient demographics. These were, standard patients with urothelial carcinoma you see in a clinic. So the median age was 70, ranging up to age 88 years old. A quarter of these patients had liver mets, a quarter of these patients had bone metastases, and only one patient had progressed on just one line of therapy. A majority progressed on 2 or 3 lines of therapy. So in order to get on the trial, a patient had to have progressed on both platinum and immunotherapy or progressed on one line of therapy and be ineligible for cisplatin. So patients were given EV and SG at different dose levels. So dose level 1 was at SG at 8 mg per kg with EV at 1 mg per kg, and then dose level 2 was EV 1.25 mg per kg with SG 8 mg per kg, and then dose level 3 we actually used full dose EV and SG together, right? So SG 10 mg per kg with EV 1.25 mg per kg. Drugs were given day 1 and 8 of a 21-day cycle until unacceptable toxicity or progression. And so we looked. First step, right? What's the maximum tolerated dose? That's the primary endpoint. So 9 patients enrolled at dose level 1, 8 patients enrolled at dose level 2, 6 patients enrolled at dose level 3, of which 5 were valuable for DLT assessments. So in dose level 3, there's actually 3 DLTs, 1 neutropenic fever, 1 mucositis, and 1 delay of therapy. But when you look at dose level 1, there's 2 DLTs, both neutropenic fever prior to adding G-CSF, and dose level 2 is actually 1 DLT, which actually probably doesn't delay the therapy because delayed in therapy due to pembroke colitis. So per the Bayesian design, the maximum tolerated dose can't have 1 DLT in 8 patients. So the maximum tolerated dose is actually dose level 3. So full dose EV and SG. So what that says, wow, we can actually give the maximum tolerated dose of EV and SG together would be the same dose used as monotherapy, which I think is the first step. Looking a little bit further at the details, when we enrolled the first 6 patients, we didn't allow prophylactic G-CSF with cycle 1, but subsequently we did, as we had 2 neutropenic fevers. And so majority of the patients after the first 6 did get G-CSF. So I think the caveat, the maximum dose is probably with G-CSF support. What about toxicities in general? So when we look at the toxicities, there were really no new toxicity signals with a combo compared to monotherapy. Diarrhea, anemia, neutropenia were sort of high up there, but we didn't see any increase in neuropathy or any marked increase in SG when you look compared to monotherapy. So all that together, what should be studying further, right? So the recommended phase 2 dose isn't actually the maximum tolerated dose just because the max tolerated dose doesn't mean that's what we need to give everyone. And so as well, I'll talk about the efficacy was quite remarkable across dose levels. But when we specifically look at just safety, the median number of cycles received at dose level 3 was only 1. So most patients quickly dose reduced. Dose level 2 patients went on to get multiple cycles. And given that EV has phase 3 level 1 data, we really want to optimize that EV dose. So that's why we chose dose level 2, 1.25 with 8 mg of SG. Question is, does it work, right? And so small numbers, 23 patients valuable for response, but of those patients, 20 to 23 had any degree of tumor shrinkage on their scans, correlating with objective response rate that was confirmed of 70%. 3 CRs, only 3 patients had PDS response. Multiple patients are still ongoing. 9 of 23 patients enrolled in the trial have responses ongoing with a median follow-up of 15 months. So I think it's really exciting. This is the first trial in any malignancy to show that 2 ADCs can be given safely together. I think at a maximum tolerated dose, that was actually full dose of each agent, although that's not what we recommend studying in future studies. I think that neutropenia was the main issue. Prophylactic neoadjuvant should be indicated per guidelines, but I think it's really exciting. We have expansion cohorts in development to further study the DAD combination in the treatment-resistant setting, as well as looking to combine that with IO in what we're calling the DADIO cohort of EV, Pembro plus SG.